Hanno già caricato le presentazioni, adesso vediamo chi sono, tiro fuori l'elenco.
Siamo a truppe ridotte questa mattina. Il fatto è che quel tipo di comunicazione non lo danno in mano per dirti a noi, a Spock, gestisce il centro.
Appunto. No, ma il centro ha una serie di problemi. Dai. Svegliati. Dai. Eccolo. Ok.
Ci siamo?
Sì. Allora abbiamo sessione 3, giusto? Monitoring 3. Ok, allora monitoring 3. Stamattina abbiamo Naomi Massaccesi, ottimo. Hai già caricato la presentazione? Ok, poi Alessio Polvani, super. Rosa Ranalli, sì. Jorge Sanchez Navarro remoto. Ok, va bene. Anna Piquet, molto bene. E basta, questa è la sessione di stamattina con gli speaker. Se tutti avete già caricato, vi ricordo che avete una decina di minuti per presentare, poi se siete nei tempi 1 o 2 domande. E alla fine della sessione poi ci sarà un coffee break e ci sarà la sessione poster. Quindi Naomi, prego.
Questo?
Sì.
Ok.
C'è anche un pointer, credo.
Sì. Good morning to everyone. My name is Naomi Massaccesi and I'm a PhD student. My project proposal is about identify sources, sinks and pathways of microbial pathogens and antibiotic resistance in aquaculture environment and aquaculture settings. So moving to the introduction, we know that the global exponential growing demand for aquatic food over the last 10 years has resulted in a shift from extensive to semi-intensive system in order to maximize the speed of growth and obtain greater yields. However, this intensification of production and high stocking density have led to several consequences, such as an increased nutrient pollution and suboptimal hygiene condition, resulting in pathogen outbreaks and the consequent overuse of antibiotics. Antibiotics are natural or synthetic substances used to kill or inhibit the growth of bacteria through different mechanisms. Normally, they can be used like prophylactic, metaphylactic, or therapeutic agents, or like growth promoters in both animal husbandry and aquaculture settings. And they usually mix with the feed of animals, or they can be applied directly in the aquaculture environment. But considering 80% of these compounds are not absorbed by the gut of fish, this compound can be released into surrounding environment through, for example, the fish phages, and these make the fishing farm a potential source of antibiotic resistance. But it's not only a source, represent also a sink, given that land, rivers, sewage discharge, or other sources for human activities may lead to the introduction of antibiotic residues in the aquaculture environment. However, we have to say that the list of the approved antibiotics, as well as the low quantity, vary across different geographic areas. But in Mediterranean area, the most commonly used are represented by oxytetracycline and quinolone drugs. The primary aim of my project proposal is to understand through a cross-disciplinary approach, mainly based on molecular microbiology, chemistry, and biosensor-based application, how antibiotic resistance and pathogenetic bacteria is introduced in open and closed aquaculture system, so referring to mariculture and recirculating a fish farm. To this aim, the analysis of antibiotic-resistant contamination is being performed in environmental samples such as seawater, sediment, and provide feedings in order to understand in which environmental compartment the selective pressure exerted by antibiotics is more effective. In farmed fish, in this case, sea bass and sea bream, in order to clarify how the antibiotic resistance is transferred into the final food products and hypothesize the risk for the human food consumption. And finally, obtain information of how microbial community composition change in response to possible seasonal and or temporal variation. In this case, the seasonal sampling is only refers to mariculture. With specific regards to my activities, antibiotic resistance and pathogenetic signature is being performed, analyzed through the short gamma taxonomic sequencing, as well as qPCR application on selected antibiotic resistant genes, in order to unveil the information on presence and levels of antibiotic resistance in both open and closed aquaculture system. Instead, the functional profile of sediment microbial communities were characterized by the use of biolactic assays. Moving to the activities performed, both mariculture and RAS sampling campaign were completed. In particular, the mariculture sampling campaign were performed on October and March in a fish farm located in Porto Venere, Liguria. This fish farm is characterized by 30 floating cages containing separately sea bream and sea bass. And we selected a different sampling site along a pollutative pollution gradient. In particular, Castagne, that is a negative control, was a beach close to a gravel beach located away from pollution sources. An intermediate site called Pontile that is located between the coastline and the fish farm. 4 fish cages located inside the fish farm. And finally, a semi-submerged channel called Canale that was mainly influenced by tidal variation and urban discharge. The RAS sampling campaign was performed on November in a land-based fish farm located in Orbetello, South Tuscany. This fish farm is characterized by 45 cages containing sea bream and sea bass separately. And also in this case, we selected different sampling sites, in particular 3 located outside the fish farm, a negative control that was a beach, a lagoon characterized by a channel connecting the lagoon with the sea, and this channel mainly influenced by the wastewater treatment plant. 7 sampling sites located inside the fish farm that was a sea bream and sea bass cage in which groundwater and recirculating water are mixed together. The recirculating water inlet and outlet in which there was characterized by water entering and leaving the aquaculture plants before or after the chemical and physical processes. And finally, the groundwater and recirculating water tank inlet characterized by the water entering into the breeding tank. All the DNA was extracted from all the samples collected in both mariculture and RAS fish farm. And also we performed the characterization of microbial community in mariculture sampling. An aliquot coming from the DNA extracted have been sent to the external sequencing services in order to do the short gamma taxonomic sequencing, and the second aliquot was used to perform qPCR analysis. Moving to the preliminary result, I investigate the presence using the qPCR in collaboration with the CNRs of Verbania of 3 antibiotic-resistant genes, in particular the black TXM, RMB, and SUL2. Also the class 1 integrin integrase gene and the 16 RNA gene in order to normalize the abundance of all the accessory genes. Observing the graph, we can say that there is no clear trend of contamination. Observing the first genes, the black TXM, show a no clear trend considering all the tested genes, instead of the Intel 1 and SUL2 that was more higher in environmental samples such as seawater and sediment instead of the gut of fish, in which RMB show a higher abundant, and this result represents interesting findings considering RMB encounter resistance to macrolides, lincosamides, and streptogramins that was mainly used in the first part of the pandemic COVID for the human use. Moving to the characterization of the sediment microbial community in terms of functional properties, we selected 3 sites in this case, so the negative control, 1 cage, and the must pollution sampling site. And we can see that below the fish cage there was a higher heterotrophic respiration, a higher metabolic potential, and also a higher functional diversity. So this means that below the fish cage we have a higher availability of organic matter, a microbial community prompt to express metabolic functional pathways and characterized by a prompt degradative activity. These results will be compared with the results coming from the RAS fish farms, and the qPCR data will be analyzed under the light of short-gamma metagenomic data that are being processing in order to provide a step forward to understand antibiotic resistance and the spread of pathogenetic microbial species in both mariculture and RAS fish farms. As a result, it is expected that the outcome of this project are going to have a very positive impact from a better understanding of this topic to the improvement of the fish farm practice in order to obtain a development of this important food sector. Thank you for your attention.
Thank you, Noemi. We have time for a couple of questions. I have one. On the fish, or the microbiome of fish, where did you sample the
Okay, I sample from the skin, the gut, and the muscle.
You know that gills are the real interface between environment.
Yeah, we also sample from the gills, but referring to the qPCR analysis, we obtain that skin and also gills and some muscle show a higher inhibition. So for the qPCR data, we eliminate this type of sample, but we hope for the short metagenomic sequence data to obtain different results.
Hi, sorry, in slide 14, the last plot, how did you measure functional diversity? That you said that...
The functional diversity, yeah, applying the biolog TMSAs. So we use different ego plates containing different carbon sources. We use 31 carbon sources belong to 6 major classes, for example, phenolic compounds, carbohydrates, and in this case was based on a common base principle. So we observed the reduction of the respiration sensitive ditretazolium to formazan, and we use the spectrophotometric at an absorbance of 590 nanometers, and then we obtain the result.
Okay, thank you. Thank you for the presentation.
Okay. If There are no more questions. We can move on. Thank you, Naomi.
Thank you.
Next speaker is Alessio Polvani, Novel Citizen Science Method for Freshwater Quality Monitoring.
So good morning, everyone. I'm really pleased to be here and to present to you guys. Today's topic will not be very focused on biodiversity. I will talk about novel citizen science methods for freshwater quality monitoring, but you would see that it is kind of related in some way. So just to begin, a brief introduction of what are the mission of me and my teammates inside NBFC. So our mission is to implement citizen science project to involve local communities on biodiversity monitoring and conservation projects. So we came up with this idea to create CS for Rivers. There's the QR code you can scan to just go to the website and check out for some more information. So CS for Rivers is a riverine ecosystem monitoring project which uses a transdisciplinary approach. The core of the citizen science is that volunteers which are into this project carry out these monitoring activities in an autonomous way. So to begin, we just took a couple of case studies, which are our pilot projects, which are on the Ombrone and Idice river basins located in central Italy. Those are the activity that we carry out in CS for Rivers. We have different activity monitoring, so this is our transdisciplinary approach. And me, as a chemist, I'm in charge for the freshwater chemical quality activity. So I would talk about this methodology and how we carry out this methodology inside CS for Rivers. So first of all, let me introduce Freshwater Watch. Why am I talking to you about Freshwater Watch? Because this is a global citizen science project who aims to monitor all the freshwater bodies all around the globe. It has so far like 40,000 samplings collected since 2012. And it is a good methodology because it is very simple and well established, and so it fits very fine for a citizen science project. So there you can see the toolkit that citizens receive during their samplings, of course, upon training sessions. The things that are monitored with this methodology are the concentration of nitrates and phosphates, also called nutrients, and the level of turbidity. We use very simple methods because we use a colorimetric reactions that everybody can do even if they do not have an academic preparation. And the data collection is very simple because citizens are required to fill a form through a smartphone application, and they will be asked to answer about general information like ecological and hydrological parameters about the sampling sites, of course, and then to carry out the chemical test and the turbidity test by using the Secchi tube. And all the data are uploaded into a common platform, which and an open database, of course, which you can find into the Freshwater Watch website. So, talking very fast about some preliminary analysis result that we had so far. So, I took just one case study, so the Ombrone River Basin, which is located, which is included in the Siena and Grosseto provinces. So, we're talking about Tuscany. I reported a GIS map which described the land use classes just to contextualize the territory where the whole project is carried out. Well, mostly we have that the land use is covered by agricultural areas and wooded and grassland areas. So it is not a very urbanized area. So far, we collected 108 samplings. Volunteers collected 108 samplings by surveying 14 different water bodies. Of course, they are all part of the Ombrone River Basin. And we have about 20 active volunteers in the monitoring, and we involved also secondary schools. So I said before that we are going to monitor phosphates and nitrates, which are nutrients. And why we do that? Because they are responsible for the eutrophication process. So we're talking about algal blooms, and these are not very good if they're present, also for the freshwater biodiversity, and it is a limiting factor for the whole aquatic life. So here you can observe a couple of graphs about the average concentration of nitrates and phosphates of the 6 most monitored water bodies, included the main river, which is the Ombrone. And you can observe, for example, that the Tressa stream has a very high nitrate concentration, and this is a significantly anthropic impact on this river, because the Tressa stream flows crossing Siena, okay, the city of Siena. So it is more subject to an anthropic impact, and so to the release of nitrates and phosphates inside the water body. While, for example, Merce and Trasubia stream are not very urbanized okay. So going forward, here we measure also the turbidity, which is an optical factor. So it describes the cloudiness of the water, and it depends on the presence of suspended particulate. So turbidity, it is a limiting factor for aquatic vegetation because it scatters the sunlight, which cannot cross the watershed, and so aquatic vegetation cannot receive the lights from the sun. And here in this case, we have a high level of turbidity on the Ombrone and Orcia rivers, which are not dependent on the anthropic factors, and not even from the color of the water color, okay? But it's mainly due to natural characteristic of the river. So the Ombrone has a high transport of sediments by itself, so naturally. So how can we assess how can we improve the measurement of nitrates and phosphates that we carry out? Because we are using colorimetric charts to determine the concentration of these chemicals. But how can we improve the quality, the resolution of the data that we have by keeping the citizen science principle? So by keeping low cost, by keeping a principle of easy to use. Well, me and my environmental spectroscopy group at the University of Siena have thought about developing a DIY, which stands for do-it-yourself, low-cost spectrophotometer to analyze the colorimetric reactions that we use in freshwater water, just to improve the data quality. And so just to have a punctual result better than a range of concentration. So this spectrophotometer allows for relies on the Beer-Lambert law, which relates the light absorption of the analyte strictly related to the concentration of the solution, and it works on the visible range, so it allows for the low-cost components use. And here there's a photo of the prototype. We have a black PLA 3D printed case, so it's very basic, do-it-yourself, because we designed by ourselves the case, and so we have an LED as a light source, and in the middle, there's the sample holder, in which the cuvettes must fit in with the analyte. And then we have a light-dependent resistor as detector. And everything is ran by a Raspberry Pi Zero W, which is a very low-cost single-board computer, and allows us to be very flexible during our measurements. So we also tried for a comparison about our spectrophotometer, portable spectrophotometer, and one from a laboratory. And we just tried to measure the calibration curve, and we obtained high accuracy and precision. So just to conclude, we succeeded in the implementation of a low-cost citizen science project. We are also able to provide additional data to support environmental local agencies, and this is because with this methodology we are able also to monitor those streams which are secondaries and which are not very monitored by the local agencies. And we are also integrating this national monitoring into the United Nations Sustainable Development Goals. Of course, the 0.3.2 regards to provide the proportion of the good quality status for freshwater quality water bodies all around the globe. And the future perspective of my work is to upscale this methodology to other contexts, hopefully in other national contexts, and to demonstrate our optical sensor on-field. So I want to thank the whole CS for Rivers and BFC team and the guys from the environmental spectroscopy group. So that's it, and thank you for the attention.
Thank you, Alessio. Questions, comments? We have time for one, please.
Good morning. I was wondering, I'm not an expert in river ecology, but since you said that the high levels of nitrogen could be from the city of Siena in one of the rivers, and you also said that a fair amount of the land use was agricultural. I was wondering if also the agricultural practices were a cause of a higher level of nutrients.
Thank you for the question. And I remind you to this slide, if you look, it's quite small, but if you look at the image, nitrogen and phosphates are mainly found in fertilizers. So, of course, the leaching of these two chemicals, which are very persistent, during precipitation, we have the runoff of this fertilizer inside the water body. So the agricultural lands are how can I say, one of the reasons of the increase of these chemicals inside the water bodies, at least the concentration.
Did you know if any of these rivers cross like a big fraction of agricultural land, so that could be there could be a bigger impact on these rivers?
Yes, most of them, because at least the half of the Umbrian River Basin is covered by agricultural areas. And, well, with our methodology, we just monitor one sampling site each month. So we want to see each month how that sampling site change. But it depends on the choice of the sampling site, because maybe it can be on an agricultural area or in a wooded area. So we do not... One of the things that I would like to do is to map in a spatial and temporal way all the river basin, but we need like 1,000 of volunteers, and this is not possible, but this could be the ideal way to carry out a citizen science project.
Thank you.
Thank you very much.
Thank you, Alessio. Next speaker is Rosa Ranalli. Title is Monitoring Pollinators to Enhance Biodiversity in Italian Cities. Thank you.
Good morning, everybody. I'm Rosa. I'm in the second year of my PhD in Milano Bicocca, and my research focused on pollinators, especially wild bees in urban areas. So I think everybody knows that pollinators are very fundamental for our life, for agriculture, but it has a very strong effect also on our health. In Europe, and we have almost 2,000 of wild bees, which are the main pollinators in Europe, and in Italy we have the half. While in tropical area we can see also pollinators such as some lizards and some small mammals. But as we know, these pollinators are very strong in decline for anthropogenic reason. And my research focused on the land use of our landscape, and especially in cities. It is because some elements in our cities can serve as refuges for these pollinators. So the aim of my research is to understand how the city can support pollinators and how the ecosystem service function in this landscape. So to this we have different sampling sites. In Italy, last year I monitored 6 Italian cities, almost the major Italian cities, and I have also sampling sites in Milano, in different places, and I sampling pollinators with different techniques. Some active techniques like the sweep net, the end nets, and some passive like the pan traps or other different traps. And I also have monitoring pollinators with observation of the behavior. The main techniques that we use is obviously the identification of these pollinators with integrated taxonomy, so with morphological analysis and also with DNA barcoding. But to understand the ecosystem service, we analyze also the pollen in the body. We wash the bees. And we have different analyses. We count the pollen grains with automated counter with ImageA, which is a software, and Eurocold Lab, it's a collaboration with another laboratory which have this multi-sizer. It is a machine that automatically counts the pollen grains. And then we have DNA metabarcoding analysis to understand exactly which plants are visited by the pollinators. So I show you some preliminary results. These are 4 of the 6 cities that I monitored last year. As you can see, we have a different number in the different colors of the column are different genera of bees. And you can see how they are different slices in a different urban gradient. We have from a small areas, urban areas, to a bigger one, and across fragmented area of insert in the urbanization matrix. This is obviously a very preliminary result, and we have to integrate this data with the landscape analysis. But you can see that in the middle part of the graph, we have the measure biodiversity of the bees in all these 6 cities. Here you can see 2 networks between, obviously, plants and wild bees. In 2 different areas. This is 2 areas of Milano. On the right side, we have a very complex network. This is a very large area. It's our natural areas. It's a control. And you can see that we have high diversity in bee species and also in plants. And it's very complex, while in a very small area on the left, very fragmented and very in the city center of Milano, you can see a very simple network. So it suggests us that we have to enhance biodiversity and protect pollinators in our cities. We analyzed also the relationship between the pollinators and the forest in urban areas. It's because some bees have the nest in the wood, in the dead wood, but also in the forest. And we see that with an incrementation of the number of the logs, woods, and some elements of the forest, we have also an increase of pollinators. When we analyze the pollen grains, so to support the pollination ecosystem service, we see that the bees with ventral scopa, so they collect pollen with the ventral part of the body, they collect more pollen than the other ones, and these bees are also the bees that have nests in the wood, in the cavities. So it's fundamental to preserve our forests in the cities. Then we analyze very polluted areas. There are two ex-railway stations, and they have a very high pollution, especially in metals, in the soils. But we find a collaboration with our botanical colleagues, and we see that we have a very strong percentage of alien plants, or naturalized alien plants. And these plants can support a very high biodiversity in our pollinators. So it was very, very strange because it was a very polluted and rich flowers. But we found a very high biodiversity in pollinators. So it's a suggestion more to support our pollinators and to enhance the biodiversity in our cities, also in polluted sites. So, these years, I start my field activities, and we have some sampling sites in Milano and in Roma also. We have planted some flower strip with some fungals to enhance the physiology of the plants and to support the trophic resource, not only for pollinators, but also for multitaxa biodiversity, so for small mammals and bats and birds. And we put also some nesting sites, and we want to test if this simple biodiversity plot can support really the biodiversity in cities to suggest it to local authorities. So I want to really thank my supervisor and tutors and my master students, and also some students that are here today, like Anna and Claudia, which helped me to monitoring activity last year.
Open for questions. Francesco.
Thank you very much, Rosa, for the presentation, and also it seemed huge monitoring and sampling effort, so really good job. And I would like to know if you used some metrics to quantify the complexity of your network between that you show in the presentation between pollinators and plants, in order to really get an index of how much diverse and complex are these networks.
So for diversity, we use the indexes like Shen or Simpson, but we want to apply the network analysis also to understand the complexity of the community. So with nestedness, for example, and also to understand how much are they stable in our landscape, because this is a sampling monitoring of 1 year. We have 3 months, but they are only 1 year. So it's like a photo of a moment. And we want to continue the monitoring, but we can try to understand if this community is stable only in 2 years or 3 years of monitoring. So we'll see at the end of my PhD.
Okay, but so you want also to study the dynamic of the...
Yes. The stability of the community, the nestedness of the pollinators, so the stability of the community, but we are very interested in the relationship between plants, because in cities we have a lot of ornamental plants, and we want to understand if ornamental plants and alien plants. So we want to understand if these plants can really support our pollinators, our native pollinator, or if they're supporting alien pollinators. We have two alien bees in Italy, and we want to understand if they are increasing with alien plants, with ornamental plants, or not.
Okay, thank you very much, really interesting.
Okay.
Very nice.
Thank you, Rosa, very much. We have now a contribution from Jorge Sanchez Navarro, and is from remote. He's connected, is he? Jorge, are you there?
Hello?
Hello.
Hello, Jorge.
Check. Hello?
Good. We hear you. Do you hear us?
Okay.
Oh, here you are. Good. You can start your presentation. Welcome.
Thank you. Thank you for allowing me to do it remotely. Can you see it?
Yes. Go ahead, please. Very good.
Hello, everyone, even if I cannot see you. I'm Jorge Sanchez Navarro. I'm a PhD student at the University of Sassari, and I'm going to explain to you a little bit my project, my PhD project. I'm in the second year already, and it's regarding monitoring and modeling of pathogen spillover from honeybees to wild bees. First, I'm going to start with a brief comprehension of what pathogen spillover in bees means. So pathogen occurrence in the wild, in wild bees, increases when in the presence of honeybees. This is due to the honeybees being a social species, and therefore they are more prone to carry pathogens like viruses and fungi. But actually, the long-term effects of pathogen spillover from honeybees to wild bees has not been studied enough. It's still unknown in wild bee communities. For that matter, the main objective or aim of my thesis is to monitor and develop models for pathogen spillover from managed honeybees to solitary and social wild bee species. For that, we've developed a methodology for which we've divided our target species into 3 groups, which are Apis mellifera, the honeybee, bombus, the bumblebees, and solitary wild bees. The sampling sites were selected according to the apiary or hive density found in each area. As you can see in the map, there's a heat map where the green areas represent the low density of hives area, and the reddish orange areas represent the higher density of hives in that area. So the first year we started with 6 high density of hive sites and 6 low density, and we are currently completing the experiment with 20 more, 10 high density and 10 low density. The transect is 250 meters long, and it's divided into 5 subunits of 50 meters. The bees within the transect are counted separately for each subunit during a 10-minute walk. So in total, it's a 15-minute walk. The walk takes place once in the morning and once in the afternoon. After the counting, we can start with the sample collection, sorry. And we aim to obtain at least 20 individuals of each group. These are then preserved in refrigerated bags to be later taken to the lab and stored in a -80 ultra freezer inside of RNA later fill tubes for later assessment in the lab. As for the flower part of our methodology, we carry out an assessment that consists in a coverage estimate using 1 square meter plots that are randomly placed across the transect 5 times per subunit. Also, for each transect, we collect 15 flowers from 3 selected species for later analysis in the lab. Said analysis for both the bees and the wild bees will take place in the Martin Luther University in Halle next year. With the gathered data from last year, we were able to test 2 preliminary hypotheses, which are wild bee species and wild and bumblebee presence is affected by the presence of honeybees, and bee presence is affected by plant coverage. As we can see here, the first results show a difference in the presence of the 3 bee groups when the observational data is compared from high densities of hive sites and low density of hive sites. In the high density of hives, we can see they show a prevalence of honeybees with a lower presence of wild bee species and especially bumblebees. As for the low side densities, the data shows a different result. With lower presence of honeybees, both the number of wild bees and bumblebees rise. The next diagrams show the relation between the mean bee abundance for each day or each site with the flower coverage found in each of the sampling sites. It shows somewhat of a trend, as you can see, but with more data, it could be properly assessed later. Finally, last year I spent 2 months in the University of Halle, where I was able to start screening and gather some very early results. I managed to analyze 4 sites fully and screen for viruses on honeybees. The viruses displayed in the graph are the black wing cell virus and 2 types of deformed wing virus. As you can see, the prevalence of the black wing cell virus is 100%. It was present in all of the sites, whereas the deformed wing virus had a prevalence of 58%. We chose these viruses because they have been recorded as the most abundant among several bee species throughout the island of Sardinia. And for the moment, there are no significant differences between high density of hive sites and low density of hive sites. But as I said, we're currently working on it, and next year, starting in October, not this year, sorry, starting October, I will be carrying the complete virus screening, and then proceed with the gathered data, proceed to do the modeling and statistical part. Thank you very much. If you have any questions, I will gladly answer them.
Thank you. Very, very interesting. Do we have question for Jorge? No questions so far? Okay.
Jorge, just a curiosity, which kind of disease bring about the pathogens that you are checking for the presence?
So the deformed wing virus, basically, it's self-explanatory. When they get it, they're unable to fully develop the wings, and hence they have problems flying or cannot really move through the air. And the black wing cell virus affects only queen bees. They are, how can I say it? They are not fully developed, and they cannot really continue the hive. For that matter, they cannot really lay eggs or properly act as a queen bee.
Well,
those are the two that we've assessed for the moment.
But the presence of virus means that the bee is definitely affected by the virus in terms of functionality, or just it's there, the virus, but not necessarily...
I don't know the percentage of, I will have to look into it, the percentage of, I mean, these results show only the presence of the virus. I don't know what the percentage of sick, let's say sick bees that carry this virus is. I don't have the data.
Well, that's an important point, because virus, I mean, if you test each of us, we do have a virus of a different kind, but not necessarily we develop the pathology.
Yeah, but again, since this is the transmission to wildlife, it's only an assessment from cattle. Let's say bees are considered cattle, and cattle pathogens being transmitted to wildlife. Yeah. So that's, but yeah, it could be, it is important to know the percentage of affected specimens.
Yeah, at the same time, Olga, you know, maybe if the animals are sick, do you consider the fact that maybe they are easier to catch? And so your sample maybe has a high percentage of infected animals just because they are sick.
I don't know about that, but could be the case. Could be the case. We usually, when we sample, we usually see them in the flower eating or pollinating, and we catch them there. Could be the case that they're easier to catch because they cannot
Thank you.
Fly well, or in this case, fly well. But it's not common that we find bees with deformed wings. Usually we find them, even if they're, like in the case of the deformed wing virus I'm talking, even if they're positive with it, we haven't found many bees. I don't think we found any honeybees with deformed wings per se. Maybe a couple, but not the majority of them.
Okay, thank you.
Thank you, Jorge. Thank you.
Thank you.
Now we can move on. And next speaker is Anna Piquet, please. Changes in foliage dwellings, spider communities along an urbanization gradient.
Good morning, everyone. I'm Anna Piquet from University of Torino, and my PhD focuses on the response of the spiders communities to urbanization. So, humans alters the environment in many ways, but urbanization is surely one of the most impactful end users. As the urbanization rate grows, there are several kinds of alterations in the environment, such as habitat fragmentation, different kind of pollution, or temperature alterations. Urban environments share some characteristics in all the words, so for this reason, it's possible to talk about urban ecosystem and extend the results that you could obtain at a local scale to a more general context. The attention towards this topic is increasing, and as we know, one spoke of this center is strictly dedicated to urban biodiversity. Inside these spokes, there are many groups working using different model animals, and my PhD and my group specifically works on spiders. I'm actually working on 3 case studies, investigating different compartments of spiders' urban communities. In the first case, I sampled the so-called foliage-dwelling spiders, that are the spiders living on the lower branches on the trees or in the shrubs. In the second case, I sampled the spiders living in the ground. And in the third case studies, I'm working on the web weavers. The general aim of all these studies is to study the variation of the community in terms of taxonomic and functional diversity along an urbanization gradient. So, in the first case study, I sampled in 32 sites along an urbanization gradient in the city of Torino, and also in a control area that is a natural park located just out of the city. I sampled in 2 seasons, and using a beating tray that, for the one who don't know, I also didn't know until last year, it's this kind of tool. You shake or beat the lower branches on the trees, and the specimen fall in this kind of tray. Then you collect them and store them in alcohol. Then I bring them to the laboratory and identify the spiders at species level.
Eccolo qua.
It was quite interesting. Okay. So, as a proxy of urbanization, I use the percentage of impervious surface. I draw a buffer with the area of 1 kilometer around each sampling point, and then I extracted the percentage of build-up areas inside the buffer. Here you can see the taxonomic response, so in terms of richness and abundance. On the left here you can see the regression along the urbanization gradient. There is a significant decrease both in terms of richness and abundance, and the 2 different colors correspond to the 2 different sampling seasons. On the right, the box plots regards the comparison between the city and the control area. Also in this case, it's evident that there is a response. There is a significant decrease. Here you can see the functional response. So we asked ourselves, does this taxonomic decrease at increasing urbanization correspond also to the loss of some functional ecosystem functions? We divided the spiders in foraging guilds based on the type of hunting strategies, and then we calculated the number, how this number of guilds varies along the gradient. Then we also analyzed the behavior of a very specific group, that is the one of the spiders feeding on pollinators with this very specific diet. Also in this case, you can see that both along the urbanization gradient and in the comparison between urban and control area, there is a significant decrease at increasing urbanization. So we can conclude that in the city there is a loss of number of individuals, number of species, and also a shift towards more generalist strategies, as the city appears to favor the ambush hunters. This homogenization could be seen as a possible results of the habitat loss. The city is simpler, is less complex habitat, and is less rich of ecological niches, so it favors that more generalist spiders. And the spiders we found in the city are actually a subset of the spiders community you can find in the rural areas. As the European Restoration Law suggests to increase the percentage of the green areas in our European cities of 3% in 2040 and of 5% in 2050, I tried with QGIS to increase the green areas in Torino of the 3% and 5%, and apply my model and see how biodiversity responds to such an increase. It's interesting to see that such a small increase like the one of the 3% that you can see in the middle figure already has some significant impacts on biodiversity. Another case study I'm working on is the one regarding the ground-dwelling spiders. I sampled using pitfall traps and following the grid that is decided at national level by the Politecnico of Milano. Here you can see the sampling effort in all the city. I didn't do everything alone. In Milano, there was Rosa, that spoke before, in our group helping, and in Roma and Firenze, another PhD student. So altogether, we managed to do a big sampling effort and sample in all these cells for 3 replicates using the pitfall traps that you can see in the bottom. This grid is in fact a trade-off between the fragmentation of the habitat and the impervious surface. I'm now sorting and identifying spiders at species level in this study, and the future step would be building a functional hypervolume of the spiders at community level, the comparison and integration with data coming from the other cities, and also, if possible, the integration of our data with multiple taxa data from other research groups. So in the last case study, I just started with it. I just finished the sampling 2 weeks ago. And this is me sampling in a roundabout in the city. And I'm collecting spiders with their webs and with the preying remains in the webs along an urbanization gradient. What I want to do here is analyze the prey remains and understanding more about the spider diet in urban environment, building a sort of trophic web. I also would like to measure temperature and light pollution along the gradient in order to involve also these covariates in the model. Thank you for your attention, and that's all for me.
Already we have a question.
Hi, I'm also working on urban environments with your same grid cell sampling design, and I'm working on mammals. So I usually use a 1 kilometer buffer for medium-sized mammals, and I was wondering, isn't it too much for spiders? Like, isn't it too big?
Okay, I also thought this at the beginning, but then I tested with different buffers, 100, 200 meters, 500, and 1 kilometer. And the 1 kilometer buffer, it's always the best ones in terms of response. And that could be because spider actually, when they are juveniles, they fly, they do ballooning. So they respond better to using a bigger buffer. I was also surprised, especially for the ground-dwelling spiders, because they are quite big. They fly just in the very early stage of their life, and then they don't move so much. But
Okay, thank you.
Oh, 4 questions. Okay. It's the last call. We can take a few minutes.
Hi, thank you for your presentation. I was wondering, I saw that you mentioned that you want to test the urbanite island effect. So I was wondering, what do you expect from that?
so I put some data logger. I already know that there is an increase in temperature going inside the city. I don't know. There are some other studies stating correlating the temperature increase with an increase in dimension of some spiders. So maybe I will try to do something to correlate the body size with the urbanite island effect. But honestly, I still don't know.
No, it was just curiosity, so don't worry.
Yeah, probably something with the body size, because there are some studies stating that the medium length of the femur increase with increasing temperature. So it will be interesting to test something like that.
Thank you.
Hi, Hannah. Thank you for your presentation and for your work. It's more a curiosity, let's say, but how do you choose the sampling sites inside the city? There are some...
So in this case, we had this national grid that it's a trade-off between fragmentation and impervious surface, and it's the same grid that was applied in all the cities. So I went in the location that the grid says. We took the diagonal, so the diagonal of that grid. I went there and tried to identify some plots that were suitable to put the pitfall traps. In the other study, I did something a bit simpler here. I went with the bike within the city to see where there are these shrubs of Juniperus horizontalis, because I standardized for species. And then I plot on QGIS and draw the buffer. And then I chose the points that were best delineated along the gradient.
Okay, in the first case study, the really first...
Same.
The same, okay.
Really, I chose before the point to build the gradient.
Okay, thank you.
Best way.
Last?
Yeah, thank you. Thank you very much for the nice presentation. I have two questions. One is a technicality and one is more of a curiosity. And maybe I missed it, but how do you define the degree of urbanization?
Sorry, how define?
The degree of urbanization.
The degree.
Yeah.
I use the percentage of impervious surface. So I have some layers in QGIS representing the percentage, the impervious surface or build-up areas. I use both of them. And then I draw this... I draw this 1 kilometer buffer, and I extracted the percentage of build-up area inside the buffer. I use that as a proxy of urbanization because in literature it's a proxy that was already used, so I chose this. But you could use also other estimators for urbanization level, such as, I don't know, the opposite, the green area dimensions. In this case, it worked quite well, this method.
Okay, thank you. And the second question is, are you interested in spiders for any specific research reason, or is it more curiosity-driven?
It happened. It's not really, it was not really... So I have the luck.
I mean, not like it happened, I guess, but like...
No, spiders are important bioindicators because they are top predators. So they are very important in these kind of ecosystems. And then I work with one of the major taxonomists of spiders in Italy. So to go back to the speech that was quite frequent in these days about the lack of taxonomy experts, I work with this person that is very expert into spider taxonomy. So it's quite difficult also to identify spiders at species level. So I have the luck to I can ask in the next door to the major expert of spiders to help me with identification, and that's an important point.
Thank you.
This was my curiosity, and how long did you take to learn the taxonomy of spiders? But
it's very complicated.
Just no,
I'm learning, but after some months, it still happened that I cannot identify even the family of one spider. It's very difficult. Working in urban areas, at the end you have more or less the same species, but hundreds, like almost 200. So, but sometimes it happened that there is a high stochasticity, that one random spiders arrive ballooning from another region, so it's very hard.
I can imagine, at the same time you are building up another part of your knowledge, because in addition to, let's say, community and ecology study, you are learning to be a taxonomist of spider, maybe.
Little by little.
Good luck, anyway.
Thank you.
Thank you. And now we have a coffee break for your pleasure.
And then the poster section, and then we will
meet again here.
All together, also with the other section. And the idea will be to have the last round table also discussing about concluding remarks, so that after the lunch everybody will be free, if you agree.
Good.
It's fine? Okay.